Hi, my name is Alex and I'm a student intern in the Courses Lab at the Gladstone Institutes. I am presenting the project Variant Effect Prediction using Deep Neural Networks for Alzheimer's Disease. Alzheimer's disease, or AD, is a neurodegenerative disease that affects around 50 million individuals worldwide. It is characterized by progressive decline in cognitive function for which no effective therapies currently exist. The onset and progression of AD are driven by genetic factors, specifically genetic variants, which are single base pair changes at specific locations within the genome. My research investigates which genetic variants increase the risk of developing Alzheimer's and why. Genome-wide association studies, or GWAS studies, have identified thousands of genetic variants that are statistically associated with AD. However, over 90% of these variants reside within the non-coding genome, affecting how genes are expressed by altering transcription factor, or TF binding. Transcription factors are proteins that regulate gene expression and bind to DNA at specific binding sites represented as motifs. By perturbing the sequences of enhancers and promoters which are bound by sequence-specific transcription factors, non-coding variants can alter downstream gene expression. One way to quantify TF binding is through single-cell ataxic experiments that profile chromatin accessibility. Within the ataxic data, we see peaks at regions of enriched signal that are indicative of gene regulatory elements bound by transcription factors. The brain cell single-cell ataxic data used as my dataset contains 24 clusters grouped into 7 brain cell types. Because the cis-regulatory code is cell type specific, 7 distinct models are needed. In the last decade, deep learning models have emerged as the state-of-the-art models for complex pattern recognition and genomic data. In particular, I extend previous work by Absek et al. And re-implement the BPNet convolutional neural network to model chromatin accessibility. The deep shaft pseudo-backpropagation algorithm was used to interpret BPNet and identify the specific bases that the model thinks are most important for TF binding. Overall, the goal of my research is to develop machine learning models to prioritize AD GWAS variants by their likelihood of causing Alzheimer's and integrate the models into an accessible platform for other researchers to use. My research can be split into 3 main phases: neural network development, variant scoring, and platform deployment. First, I trained and tested complex convolutional neural networks using Keras on TensorFlow to learn the relationship between a genetic sequence and its TF binding profile. Each model was trained on around 200,000 data points with an 80-10-10 train-test validation split. To further improve these models, we made 2 novel optimizations: negative sampling and TN5 bias correction. Negative samples are regions of low TF binding that were added to the training dataset to increase the model's predictive range. After many iterations, I found that a 1-to-10 ratio of non-peak to peak samples achieved the best balance between prediction accuracy and range. These negative samples were also GC meshed to prevent the model from learning any confounding factors. Unlike the chip nexus TF binding data used in the original BPNet study, ataxic data contains biases that result from preferential binding of the TN5 transferase enzyme used in the sequencing process. To negate these effects, a separate BPNet model was trained purely on non-peak regions and added as a secondary input in the last layer of the main model's architecture. In this manner, the bias model predictions would be regressed out of the main model's weights. Next, to score GWAS AD variants, I calculated the Jensen-Shannon divergence, or JSD, and log-fold change, or LFC metrics, to rank the variants based on the largest variant effect, which is the model's predictive difference between both alleles of a variant. The JSD score represents the difference in shape between the predicted profiles, while the LFC calculates the difference in the total counts. Lastly, I developed a production-ready variant effect prediction platform. Given a variant and cell type, it uses the model to generate model predictions and important scores for both alleles, then calculates the difference, displaying all of these figures for the user. To assist in variant interpretation, the platform also provides a list of all relevant TFs for the given location. Overall, the CNN models achieved high accuracies, with Pearson-Spearman correlations around 0.75 and binary AUC scores around 0.9. These values mean the model accurately predicted levels of TF binding, a regression metric, and could clearly distinguish between peak and non-peak regions, a classification metric. The Pearson correlation of the excitatory neuron model's predictions on test chromosome 1 is shown in the scatter plot, which portrays the high correlation between the true count and the model's predicted values. As shown in the table, some models may have been hindered by insufficient data, and the lower performance can be attributed to data scarcity. After scoring the GWAS AD variants, we found that our models mostly agreed with rankings generated by the previous generation GKM SVM models, validating that both models accurately learned patterns of TF binding in brain cells. However, the more complex neural networks were able to increase the confidence level in certain variants and nominate new previously unexplored variants. The successful implementation of the variant effect prediction platform was also used to validate the top scoring variants. Using this platform, biomedical researchers can accurately evaluate and prioritize their variants of interest in not just Alzheimer's but all genetic brain diseases. For example, using the app output, we can see that our top-ranked variant, RS636317, reveals a clear footprint where a TF would bind in the reference allele that disappears with the alternate allele. This effect is highlighted in the log-fold change graph, which also reveals oscillations characteristic of CTCF nucleosome spacing. Additionally, the delta importance score graph isolates the CTCF motif, presenting a potential biological pathway. This variant was recently validated by Novikova et al. 2021, which revealed that the T alternate allele disrupts the CTCF transcription factor, increasing expression of the AD-implicated MS4A6A gene. Another example is RS130257717. We show that the log-fold change graph similarly has a TF footprint, and the delta importance scores isolate a KLF4 motif. The significance of this variant was validated using an MPRA experiment conducted in my lab. In the literature, the KLF4TF is known to potentially affect the BIN1 gene in microglia, a well-studied gene relevant to Alzheimer's. In conclusion, our CNN models accurately learn the complex patterns of TF binding in brain cells and identify candidate causal variants for AD, some already validated by biological experiments and others still to be explored. Our novel model architecture, combined with the optimizations for dynamic range and TN5 bias correction, significantly improved accuracy and helped us perform better than the GKM SVM models used in previous research. Lastly, the variant effect prediction platform can help researchers prioritize the variants of interest by picking out the most important needles out of a haystack for any genetic brain disease. In the future, we plan to freely distribute the platform to biomedical research labs, validate and improve the models using MPRA data, and apply our models to other neurological diseases such as Parkinson's disease or ALS. Overall, our neural networks show promise in nominating candidate causal variants for AD, and with the platform, this research can truly play a crucial role in identifying the genetic pathways that cause Alzheimer's and other genetic brain diseases.